Good morning, everybody. So today's we'll continue our discussion starting from the place where we stopped last times. Actually, I'm just trying to revise everything today. So when I'm just trying to revise, our last time discussion was regarding, the control experiment and the components of the control experiment and some of the variables actually. As you know, when we talk about experiment, we have two types. We said that is we have two types of experiment, which is the control experiment and the control group and the experimental groups actually. So while we are doing a control experiment, we have two components, which is the control group and that of the experimental group, as we see here. So the control group is a group where an experimenter is not where the experiment is an experimenter is doing his own experiment, and the control group is that is the one which is not the control group is the one which is not which is not treated by the experimenter, whereas the experimental group is the group where the experimenter is doing different type of experimental treatment. So as you see here, the control group is with no experimental treatment, and the experimental group is with experimental treatment. But the importance of having the experimental group is the control group is when we are doing a certain experiment and finally we'll reach to a certain result. So based on that result, whether that thing is coming with every type of experiment or not, we need to have some kind of control group. So if the result of the experiment is the same in both control and experimental group, maybe our experiment is already failed or our experiment is not correct. But if the results are different in the experimental group and the control group, so that we can compare that the results of the experiment will be accepted, so that only the experimental group is just giving that result, whereas the non-experimental group doesn't give that result, so that we can compare with this and we can check that our experiment is correct. So that was the purpose of having control experiment. And when you come to the variables, as I just already talked about last time, we have around 4 types of variables. We have the independent variables, we have the dependent variables, we have the control variables, and we have the confounding variables. So when you come to the independent variable, this is a variable that's changing or manipulated by the scientist. You see, for example, last times we have tried to see that the tomato seeds are not growing within the tomato juice, we said. So from that, you see, the growth of the seed of the tomato will be the dependent variable, because that is a variable that's measured or observed in an experiment, because our interest is to see whether the seed is grown within the while it was found within the tomato juice or not. So that is known as the dependent variables. Whereas there are factors, you see, that just already affect this growth. Maybe the juice or the other thing or the chemical which is found inside the juice might be considered as independent variables. And when you come to the control variables, you see, for a seed to grow, there are certain conditions, maybe the temperature, maybe the air, maybe the sunlight, and maybe the other factors that just already help us for the seed to grow, or any seed to grow. Those things can be control variables, so that they will be available as just already they are available as it was needed by the seed. But if it's not, they are available at the need of the seed. You see, due to the independent variables, the seed doesn't grow. That is just already the dependent variable. So our interest is to measure whether the seed is grown or not while it was found inside the tomato juice. So this time the tomato juice is considered as the independent variable, whereas other factors, you see, that helps the seed to grow can be considered as control variables, and these are factors other than the independent variable and that are kept constant. So these are not already manipulated. They are kept constant. They are available for the seed as it was needed by the seed. You see, but due to the independent variables, you see, the dependent variables will have some change. It might grow or it might not grow. So that depends on the independent variable. So this is known as the control variables. Whereas when you come to the confounding variables, factors that can't be controlled, which may influence the result of the experiment. You see, this is the factor that is beyond our control. For example, you can't determine whether something is, done in desert and whether it is done in, in highland. You see, this is the factor that, that can be just available in the environment. Maybe you may just change the places, you see, but if you are in Addis, you cannot determine. Sometimes there will be, in the morning, it could be hot, or sometimes it could be cold. So you can't change this thing actually. But this is beyond your control, or this is beyond your capacity to control this thing. So that, such kind of things, we call it, it is known as, the confounding variables. So the other part is analyze and interpret the results. You see, we set an experiment and we do our experiment in the laboratory, so that after we do that, we are just already collecting data, and those data will be collected, organized, and finally those data has to be analyzed. After the data analysis, the results of the analyzed data or the analyzed data will be interpreted. So analyze and interpret the data means their data are controlled, organized, and finally analyzed. After analysis, they have to be interpreted. So that is known as, the step where people analyze and interpret the results. And finally, after the interpretation, we'll just already draw our conclusion, so that we'll understand that, you see, we are just already keeping everything which is important for the growth of seeds, and we control the control variables. And after that, we have, the independent variables here, so that, after a certain period of time, you see, you might think that, how long that the seed need to grow. After that period of time, if you didn't see any kind of, sign of growth of the seed, you see, so that we can reach to a certain conclusion, or we are just collecting data. Finally, at the end, we found that, the seed is not grown. So that is just a data that is analyzed and finally interpreted. From this interpretation, we found that, the seed is not grown while it was kept inside the independent variable, even though everything are kept constant or controlled, which means the control variables are kept constant. You see, only the independent variables is affecting, the growth of the seed. Finally, we reach to this condition. So that is known as, the result. So after this result, we interpret that result that these things is not grown inside the independent variable. So from this, we draw our conclusion. We'll draw our conclusion. From our conclusion, you see, so this conclusion will give us, it is an answer for the hypothesis based on the data obtained, and we'll just already come to a recommendation. You see, so in our recommendation, we can understand that we conclude that, a tomato seed is not grown while it was kept inside the tomato juice. That's our conclusion. So from this, we just draw, a recommendation. So that why does this seed doesn't grow? So we'll set a recommendation, and we might give, some suggestion. Why does, that seed is not grown? So this is the way how research can be taking place. And finally, after we have these all things and we've done our research, we'll come to, report writing system. You see, we have to report this thing for, for other scientists, or we just let this thing for publication in order to disclose the knowledge for everybody. Because the interest of doing experiment or the interest of doing research is in order to acquire new knowledge, we say. So based on our experimentation, we have just, reached to a certain conclusion. Finally, that is a finding for us. So this finding has to be disclosed for other bodies. You see, so that is done by reporting system. So reporting results is a means of communication with other scientists, other fellow scientists. Actually, it's all, if you are doing in a standard research, there are, concerned bodies that are just already evaluating and accrediting these researches. It's not already disclosed for everybody, but there are concerned bodies that the research will be just go through. And finally, those concerned bodies, depending on their stage, they are doing their own evaluation. Even, prior to this, it's not like that of the previous scientists. The previous scientists, 2,000 years ago, during the period of, the ancient scholars, new finding will be coming through explanation. So if you don't have, any against explanation for that, we'll accept that, and that will just already accept it and stay long. But currently, that's not the way. So there is a new method that we call a scientific method. So within this scientific, if you find something, whether you do it, whether you find it through research or whether you find it through experiment, before it's disclosed for everybody, it has to be checked. You see, it has to be proved. So everybody which is interested on this group, based on your procedure, standard procedure that you said, you see, everybody will try to repeat that experiment. So they have to repeat and they have to find the same result. If they repeat that thing and find the same result, you see, then they will accept it and they will give accreditation for that. So that if your research passes through this whole stage of the concerned bodies and get approved or get accreditation, and finally they will prove that that theory or that finding is true, or they will accept that one, and finally the final publication will be given for that research, and finally official publication will be done with certain magazines or something like that. There are Nature magazines, there are also science journals and something like that. So your results will be published there, and it could be disclosed for everybody who is interested of that knowledge. So that is the way actually. So let us see something here. So when you come to this part, your research has to fulfill those three things: accuracy, reliability, and validity. So when you say accuracy refers to how precise you measured or count. You see, while you are doing your research, there might be measurement or there might be counting. You may do, you see, when you do your experiment, you may do, for example, you are doing your experiment maybe 3 times or 4 times. You may repeat that experiment for 9 times. In order to get the real value, you have to repeat your experiment again and again. So you might repeat maybe 9 times, you might repeat maybe 10 times, or you might repeat 7 times, whatever. But when you do this thing, you have to count correctly. If you just repeat your experiment 7 times, you need to show that that repetition has to be 7 times. If you are doing 1 time, 7 times. If you are doing other times 6 times, if you are doing the other times 5 times, that's not accurate. So you need to do it 7 times every repetition. So that you can find the real result. So your result has to be close to the real one. So you need to count it correctly. Or if you have a measurement, for example, if you want to measure 2 ml of water, you can measure 2 ml of water with syringe, you can measure 2 ml of water with burette, you can measure 2 ml of water with pipette, you can measure 2 ml of water with measuring cylinder and something like that. But which one will give us the correct 2 ml? So this needs some kind of training in the laboratory. So the precise and the accurate measure might be given with the better materials, and that will be the burette one maybe. You see, so we need to do our experiment based on standard procedures, and that it has to be as far as possible, it has to be accurate. So when you are doing it with a better accuracy, the result will be more reliable and more acceptable too. And the other thing is reliability. This is concerned with if you are doing your result accurately and precisely, so that your result will be more reliable. So this is concerned with repeatability and dependability. So you find certain results, and that is just drawing to a certain conclusion, and finally you may just pass this thing for the concerned body. So those higher concerned bodies accept that result, and they will repeat the experiment just based on the standard procedure that you set. So if they just do it in a well-staffed laboratory, in a well equipped professionals maybe. So if the results or the finding of the results, if the finding of the results in your experiment and in those bodies are different, so your result will be fragile and it has to be discarded. So if the result is somewhat similar, though these times it could be accepted. So that the reliability means it is the repeatability and the dependability of your result or your experiment. Okay, so that is done by using more accurate and precise measurement procedures. And finally, if you just follow this accurately, if you are doing your experiment accurately, precisely, and finally if it's reliable, finally your result will be valid. That means it's going to be what accepted. You see, when you come to validity is state of being valid or genuine. So for example, we say that a tomato seed is not growing while they are inside in a tomato juice, we say. So that we can understand that inside the tomato juice, there is something that hinders the growth of the seed of the tomato seed. That hinders. So that we just proved to this conclusion. But finally, when the experimenter, even though the tomato seed is not growing while it was inside the tomato juice, but what is the factor? Maybe if the factor is out of this juice, if the factor is out of this juice, our experiment is not valid. Because we say that there is something inside the tomato juice. You don't know, for example, at that time, you don't know which chemical or which component is it that prevents or hinders the growth of or the germinations of the seed. You see, but you are just sure that there is something, or you are sure that the juice will prevent or hinders the germination of the seed of the tomato seed. You see, the seed of the tomato. But after you say that and then draw that your conclusion like this, if someone is doing, or with concerned body, higher concerned bodies doing their result, if they find other thing other than the tomato juice, or other than the chemical that's found inside the tomato juice that prevent or that hinders the germinations of the seed, so these times your results and their results are the same. But one thing is common, the seed is not germinated, but the germination of the seed is hindered not because of the tomato juice, but because of something else that you cannot discover it. So these times your result will be discarded. So in order to avoid such kind of things, you have to be accurate, we say, you have to be reliable in your experiment, and finally, it has to be valid. So that, once you draw your conclusion, you need to be sure that, your conclusion has to be, your result has to be, your conclusion has to be based on, your results, and it has to be, correct. And you have to be sure for that. So once, if you are sure for that, and then finally, if it passes through these whole steps, and finally your result will be valid, then if it is valid, as I told you earlier, it could be disclosed for the wallet, and it will get acceptance. So when you are doing, generally we say that there is reports writing, we say, you see, while you are writing reports, and what things have to be fulfilled in your research or in your report. You see, while you are reporting this thing for your higher officials, you see what has to be followed, or what has to be the steps. So the first step is you have to write your title precisely. Then you have to write your hypothesis. Then you have to put on your prediction. And finally, you need to include, the procedures, each and every procedure that you are using. Because every standard research and procedure have to have its own procedure. Every standard experiment and research have its own standard procedure. So you have to write down those details. And maybe the apparatus you are using, the details of any chemicals used, the details of any organisms used, and details of any collected experiment results, those things have to be fulfilled. And finally, you have to put on the result, and conclusion will be set, and finally the evaluation will be done. So using this, you can report your result, and finally the concerned body will approve that thing, and finally it will be disclosed for everybody else. So this is the way how we can conduct experiment or research, and finally how that thing has to be reported for the higher official. Thank you. So now let's come to the other part. This is known as, lesson note 3. So our today lesson is the third lesson. So we'll come to a new topic, which is known as, the tools of the biologist. So today we're going to talk about, the tools of a biologist. That one is simply a revision. Last week we have just had some kind of, network problems, so that the lesson hasn't continued properly. That's why I'm just revising some of the point. So now today's we're coming to the new topic, which is known as, biological tools. So let us see those tools that are used in biology, in the field of biology. So biologists use special equipment, some apparatus used mainly in the laboratory, such as, we might use, obviously, that you are familiar for this thing, which is microscope. And we are still using, centrifuge, and there might be petri dish and measuring cylinder and balances. So these things are, materials that can be used inside the laboratory. There are other materials too that's not mentioned here, but some of the material, the main material that you might use inside the laboratories, the microscope, it could be a light microscope or it could be electron microscope. You see, again, this electron microscope can be further classified into, scanning electron micrograph, and there are other types too. So the electron microscope might be further classified based on, their nature or their properties that you are going to observe it under it. And so the light microscope, again, this light microscope based on its source, again, it can be further classified into two, which is known as, the one which use sunlight, and the others might use what you call, electrical light. But generally, they are using as a source light energy. So that we call that's known as, light energy. And here you have, centrifuge. You see, I'm not going to tell you, what the functions of microscope, because everybody knows this thing. And when it comes to centrifuge, this is something that's used to separate, solid from liquids, so that it can just already separate us with a short period of time. So by rotating, in a very high speed, so that within 2 or 3 minutes, up to 5 minutes, we can separate, a liquid from solid. So the liquid will be settled down within our materials, which is not a test tube, and whereas, the solid will be settled down to the lower parts of, the test tubes, whereas, the liquid will be floating up. So the liquid will be the supernatant, whereas the solid will be the precipitate, so that it can differentiate that. Petri dish, this is something that you can use inside a laboratory in order to do different kind of things. It's mostly when you are doing, microbiology labs, Petri dish are very important. And the other thing is measuring cylinders, as I told you earlier times, where we are measuring liquids, we can use measuring cylinders, we can use pipette, you see, we can use pipette too. We can use, burette, you see, we can use syringe. So those things might be used. So we have different things actually. You can use measuring cylinder. Even, there are also flasks that can be used. So measuring cylinder, we can say, or graduated cylinder, and there might be pipette, there might be burette, there might be syringe, and those materials can be used for measuring. And still we do have balances. So this balance can be tri-beam balance. You see, tri-beam balance, this is manual type, or there might be electrical balance. So when you come to electrical balance, the difference is their significant digit. So in electrical balance, we can have, a 0.001 digits can be measured using electrical balance maybe. Whereas in this case, we may just start counting from 1 gram, starting from 1 gram, we can measure using tri-beam balance. So balance might be there. So other apparats used mainly in the field such as, you see, the other thing can be used in the biology is we can use more of these things are used in the field while we are doing our work on the field. So we can have, the quadrants, we can have the plant press, we can have the theodolite, we can have the GPS receiver, we can have the flow meter, the putter, you see, and data loggers. Those things are known as, the apparatus that can be used in outside the laboratory, especially when we are doing our experiments on the while doing our experiment on that of, the field, we might use those things, you see. So these times you can have quadrant. Quadrant is something that can be used as while we are estimating the number of, animals found in the fields maybe, you see. So this is something that can be measured at square. So it has a width of 1 meter and that of, length of 1 meter. So that's 1 meter by 1 meter area. So the total is 1 meter square area. So within this area, we count the number of animals available there. So that is an apparatus used as in order to count or estimate the number of animals found in a certain area of land. So that is used for that purpose. And we'll see it while we're doing our field work. And the other thing is plant press. This is help us to just already, dry leaves of different trees by just already putting inside it using some procedures, for examples. So using, other materials, we try to press the leaves, and so that we just keep it for a certain period of time, and we try to make it dry by just sort of keeping its green color as it is. If you just keep it, or if you just, let the leaf to dry outside, it might dry, but it might be broken or it might change its colors. Normally it changes its color. But if you are using plant press to dry your leaves, there is no, fear of, breakage or breachability of the leaf, and there is no color change as it is, so that we can keep as it was, just keeping its color. So we can just already make, plant album, so that, we can just already help us to differentiate the leaves of different plants by just already using, using this plant album. And the other thing is theodolite. For example, when biologists comes out of the laboratory for a field trip, you see, they might use a theodolite. This is help us to just already see the areas of the slope of the area. You see, that of the engineers are using this thing. So still biologists are using in order to measure the slope of the area. They use theodolite. And the other thing is a GPS receiver. This is in order to find out, you see, in order to find out, the areas. If you are just already, goes to an areas where you don't know it before, so in which direction are you going? So you can use, GPS, so that, you can just already locate the particular area that you are concerned for, and then you can do your field work without any problem. So that's the GPS. Actually, it can be helpful, and still we do have, the GPS inside our laboratory, so you can use it. Flow meter, this is a material used by biologists in order to measure the speed of water. You see, this is other materials. This is still, there are so many other materials, but I'm just talking about, the one which is just, the main one. So the flow meter is the material which is used as in order to measure the speed of water. Speed of water will be measured by the flow meter. And here you have putter or aspiration. So when you come to putter or aspiration, this can be made inside the laboratory using a test tubes and, a glass tube. So you'd have a test tube like this. So using these test tubes, we might use a glass tube like this, so that those glass tubes will be just already prepared like this. So one of the parts here, it can be covered with some kind of, textile. Then you just prepare this as a glass. There is also, a whole cork, or what is known as, a whole rubber. There is a cover here. There is known as, glass cork cover, or what is known as, rubber cover is used here, so that you can just already prepare such materials. And then if you find, tiny insect from the burrows of soil, burrow of, walls, burrow of logs, and burrows of what's known as, the burrows of, trees, you can just already suck through one of part, you suck through your mouth. In the other part, you try to, suck the insect, and find the insect will be collected here. So in doing that, you try to collect some small insects from the burrows, burrows of holes maybe, or from burrows of, what's now cracks. It means, when I say burrow, it means crack, from the cracks of, stone, from the cracks of walls, from the cracks of, woods maybe. You try to collect, tiny animals using this material. So this is the materials. Might be we'll do it inside the laboratory and try to how that can be applied. And there might be data loggers here. So the data logger is something that you are just already writing on your records. So when you go outside the laboratory, you are just getting so many things. So you need to write down that information. Either you can write it with paper, or you might have, a tablet-like structure, so that you can put on your data on it. That's what data logger is. So you can use electronic device, or you can use, a hard copy to write down your information. So that is the way. And the other important thing is, after we have talked about this, we try to see the relevance and the promise of biology. You see? So this is biological science actually. So this biological science have its own relevance, or it has its own importance. So since biology is a science of, life, biologists undertake all kinds of research. So biological have relevance in most every aspect of life. So there are some which that are undoubtedly of a great importance. So for this, for example, biology have a great importance in case of, agriculture, you see, and still biology have other importance in other subjects too. So we'll see those things. For example, this is common actually, biology is important in agriculture. So some of the importance of agriculture is already discussed in grade 10. That's obviously not, but there on the knowledge of biology can be applied in agriculture, so that those agriculturalists they can just already obtain some advantage of biology. So they want to do some kind of research in order to prepare plants which are well adapted to new conditions. Might be change of weather there, or it might be shortage of water there, or it might be you can think of that right today's swarm of locust is a great threat for our agriculture. So if geneticists are doing their own research, and that if the plants are capable of making their own pesticides, these times the swarm of locusts can be easily avoided. But unfortunately, our research centers are not as such developed, and we don't have such a plant actually. So that's not possible. But anyways, we can just already make a plant that can just make their own pesticide, so that the swarm of locusts might not be a threat right today. But that's one of the advantages actually, to produce plants which are well adapted to new conditions, and to produce plants that are capable of producing their crops quickly to farmers and can obtain more than one crop per year. You see, actually not drop, but this times make it crop. So growing, we can shorten our the growing season of the plant. On average, right today in Ethiopia, for a complete cycle of a seed or for a complete cycle of a crops, it needs from 4 months up to 6 months. You see, on average, from 4 months up to 6 months. So the farmers needs to complete, their crops, for their crops to complete their life cycles, they need 4 to 6 months. You see, what if you make, your plant growing season for, if they are capable of completing their cycle within 3 months, you see, so that we can shorten the growing season, okay? We can shorten the growing season. So this is one of the advantage actually. Still, we do have, some types that can complete their growing season within 3 months, so that the farmers can cultivate the land not only 1 times per year, but they can just cultivate, 4 times maybe, or 3 times maybe. So this is an advantage. Rather than cultivating their land once crops per year, it's possible if you just make them, if they make it, 3 times. You see, that will be more profitable. Will produce more yield, so that will be, better. That will produce a better income for the farmers, and they will supply also great supply of, yields for the country too. So that will be a great, innovation maybe, or a great result, or a great achievement maybe for the farmers of Ethiopia if they do like that. But anyways, this is the way. You see, the biology help us in order to do some kind of research, and we can try to make planters to shorten their growing season. Maybe right today's the average time is this one, 4 to 6 months. But if you make them the plant to complete their life cycle with 3 months, that will give us, better chance in order to produce, more plants within a year. And to produce plants which are disease resistant, as I told you earlier times, swarm of locust is currently a problem. So if they are capable of using their own pesticide, that would be a great thing. And some kind of disease might result it. Maybe last year there might be disease of, coffee, so that the quality of coffee is somewhat compromised. But if you make the coffee to produce some pesticide, might be a good thing. But while you are doing this thing, maybe its organic nature might be somehow in questions, maybe. But anyways, this is the waste. The importance of biology is. And the other thing is to produce plants and animals which has better quality and quantity of yield. These are some of the advantages of biology in agriculture. And the other thing is in medicine. Some biologists are closely involved in medical work. So biologists are able to work as medical researchers, maybe microbiologists, biomedical biologists, and they may just help in development of drugs. You see, drug development research, like, biomedical science biologists and pathologists. They may serve as they may try to develop, new drugs, because by doing, different threats right today, that corona is a great threat in our world, so that, scientists of the world are trying to develop a new vaccine against this COVID-19, so that, SARS virus against SARS-CoV-2, SARS-2 virus, you see, SARS-2 virus, the virus that caused the corona disease is COVID-19 is, SARS-2 virus, so that, they are working against this SARS-2 virus to just already develop, effective vaccine. So still biologists might involve on this issue, on these areas. And specialized researchers like biomedical scientists, pathologists, etc. You see, the biomedical biologists, they are doing in vaccine production and something like that. Pathologists, they are working on cancer, oncologists, so that they try to develop new drugs that can just control this cancer, or they may just already have some kind of vaccines. Maybe, as you see, that there is known as, for those, youngs that at earlier age, they will just already taken, or they will be given, the cervical cancer vaccine. You see, girls of 13 age, 14 age, below 15 years, they will take, this cervical cancer vaccine, so that they will be protected from the development of, or from having, cervical CA or cervical cancer, because cervical cancer is caused by a virus known as, there is a virus that causes that thing. So if they do have, these vaccines, they will not develop this, or they will not be exposed to that of, this, cervical CA, so that, specialized researchers might be involved in development of, drugs and that of, vaccines. Still, this is also the area for the biologists and special research, particularly on cancer, oncology, as I told you earlier. So there are also other areas. So biology and environment. So when you come to biology and environment, so you can see that biologists are actively involved in conservation of environment from deterioration. So they play a great role in monitoring the impact of global warming. You see, right today global warming is a serious concern. You see, it's a serious concern for our world, so that some scientists, especially ecologists, and those ecologists will be just already contributing a great. You can remember that the East African ecologist Professor Wangari Maathai, she was just have a great role in environmental protections, especially she's a Kenyans, and there are also other ecologists in Ethiopia too who are just doing a lot in this conservation and ecological conservation, and they give advice to government on how to conserve the environment and induce new species, and they still concern the government how to develop new policies regarding the conservation of the environment and conservation of the natural resource. So this is the ways how they are just trying to help their country. So this is the thing that I have for today. So for those of you who have questions regarding this, you can ask your questions. ጥያቄ ያላችሁ ሰዎች ካላችሁ ጥያቄ መጠየቅ ይቻላል። እንግዲህ ለዛሬ ያዘጋጀሁት ነገር ይሄ ነው። ጥያቄ ያላችሁ ሰዎች ካላችሁ ጥያቄ መጠየቅ ይቻላል። ጥያቄ መጠየቅ ይቻላል። ይሰማችኋል? ሄመን ሩይ ይድና ይሰማል?
አዎ ይሰማል።
አይሰማም።
አሁን ባወራነው ነገር ላይ መጠየቅ ይቻላል። ባለፈውም ላይ ያለ ነገር ካለ ግርታ ነገሮች ያው ክላስ ላይ መልሰን በደንብ እናዳብረዋለን፣ ግን አትሊስት የሆነ ኢንፎርሜሽን እንዲኖራችሁ ለማድረግ ያክል ነው። ስለዚህ በእነሱ ላይም ጥያቄ ካለ ማንሳት ይቻላል። ኦኬ፣ እናንተ ማጠይቁ ከሆነ አንዳንድ ጥያቄዎችን ይጠይቃለሁ እዚህ ጋር። ኦኬ፣ ሩይ ይሰማል?
አዎ ይሰማል ሰር።
እሺ፣ can you tell me the difference between controlled variables and dependent variable? Difference controlled variables and dependent variables.
Controlled variables are
controlled variable ምንድነውና dependent variable የምንላቸው ነገር ዲፈረንሳቸው ምንድነው?
control variables are variables which receive treatment.
Dependent
And dependable variables are variables which do not receive.
which is not going to be treated.
Yes.
Okay, good. Thank you. Heaven. Zalaka.
Yes.
Can you tell us the difference between control variables and dependent variable?
Control variable is we can control it with different samples, and we can make it constant. But
Dependent variable.
Dependent variable is it depends in sample to sample. It's not constant.
Okay, it's not constant. Good. Can you tell me the difference between independent variable too? You see, now you tell me the difference between controlled and dependent, but can you tell us about independent one?
The independent one is the independent experimenters changes to see the effects of the performance at dependent and
Its effect on the dependent variable.
yeah.
Okay. You see, አሁን እየተረዳችሁት ነው ማለት ነው። ኢንዲፔንደንት ቫሪያብልስ የምንለው ቫሪያብል ነው፣ ግን ዲፔንደንት ቫሪያብል ላይ ተጽዕኖ ያመጣል። ለምሳሌ ቅድም ያወራነው አስታውስ። ቶሜቶ ሲድ ይበቅላል አይበቅልም ማለት የት ውስጥ ነው የማይበቅለው? ኢንሳይድ ቶሜቶ ጁስ። Whenever the seed is found inside the tomato juice, the tomato seed doesn't germinate. This is our interest. You see, እዚሁ ስንመጣ ኢንትረስታችን ይሄ ነው። አሁን ይበቅላል አይበቅልም የሚለውን ቼክ ማድረግ ይፈልጋል። ሃይፖቴሲሳችን አይበቅልም ብለን የተነሳ ነው። ስለዚህ ይበቅላል አይበቅለው የሚለውን ቼክ ማድረግ አለብን። ቼክ ስናደርግ ምንድነው የምናደርገው? አንዱን ሲድ ቶሜቶ ጁስ ውስጥ እናስቀምጣለን። አንዱን ሲድ ኮንትሮል ኤክስፐሪመንት ውስጥ እናስቀምጣለን። ኮንትሮል ኤክስፐሪመንት ለጊዜው ያዘጋጀነው ምንድነው? ውሃ ነው። ዲስትል ወተር እናዘጋጅና እሱ ውስጥ እንከተዋለን። አንደኛውን ቶሜቶ ጁስ እናደርግና እንጠብቃለን ማለት ነው። ከዛ ዘር ለማለት ለመብቀል የሚያስፈልጋል factors or conditions which are important for germination of seed ብለን የምንጠራቸው ነገሮች አሉ። ሁለተኛ የተማርናቸው see air አለን worms አለን water አለን። እነዚህን የመሳሰሉ ኮንዲሽኖችን እናመቻችለታለን ማለት ነው። ከዛ we'll wait ለተወሰነ ጊዜ እንጠብቀዋለን። አሁን እዚህ ጋር ስንጠብቅ ኢንትረስታችን ምንድነው? ሲዱ ጀርሚኔት ያደርጋል አያደርግም? so germinations of seed ምንድነው? እዚህ ጋር dependent variable please አዳምጡ። germination of seed ምንድነው? dependent variable. Germination of seed dependent variable ይሆንና we are going to measure whether the seed is grown or not. Where? Inside the water, distilled water, and inside a tomato juice. But our concern is the growth of the seed inside the tomato juice. So that is our dependent variable. Independent variable ነው ብለን ስንጠራ independent variable ምንድነው? ፋክተር ነው። ይሄንን growth affect የሚያደርገው ፋክተር። so ለኛ ዘሩ አይበቅልም ወይም ይበቅላል ብለን ያሰብነው ምስ ነው ቶሜቶ ጁስ ውስጥ ኮንሰርናችን እሱ። ስለዚህ the tomato juice is the independent variable. You see, the tomato juice is the independent variable, mind you. That is our concept. That is the thing that we are just manipulating it, independent variable. But this independent variable will affect the growth of the seed of the tomato inside a tomato juice. So the tomato juice is the independent variable, whereas the seed itself or the growth of the seed is the dependent variable. So the dependent variable የዘሩ መብቀል ነው። independent variable tomato juice ነው። ስለዚህ በቶሜቶ ጁስ ውስጥ ይበቅላል አይበቅልም የሚለው ነው ቼክ የምናደርገው ማለት ነው። ስለዚህ ኢንዲፔንደንት ቫሪያብል በስንጠራ that is not the tomato juice, dependent variable the seed itself. ኮንትሮል ቫሪያብል የምንላቸው ደግሞ ሪሜምበር ቅድም ምን አለ? ዘር ለመብቀል የሚያስፈልጉ ነገሮች ሙቀት ያስፈልጋል፣ አየር ያስፈልጋል፣ እንደዚህ ብለን የሚያወራናቸው ነገሮች አሉ ከዚህ በፊት አለ እና አየር ያስፈልጋል ብለናል። ሶ ሙቀቱን፣ አየሩን፣ እርጥበቱን ብለን ስናስብ እነዚህ ነገሮች ደግሞ ፋክተሮች ናቸው። ስለዚህ እነዚህን ፋክተሮች እናደርጋቸዋለን አቬለብል እናደርግለታለን ለእጽዋቱ። ሙቀት እንሰጠዋለን፣ አየር እንሰጠዋለን፣ ከዛ በተጨማሪ እርጥበት እንሰጠዋለን። so these are control variables ይባላሉ። we control this thing and we just available this thing equally for the seed which is found inside the distilled water and for the seed found inside the tomato juice ለሁለቱም ኢኳሊ available አድርገናል። ይሄን ፋክተር ሊሚት አላደረግንም። ስለዚህ እነዚህ ሪል ፋክተር ናቸው ብለን ስላሰብን እነሱን available አድርገናል። አየር ሰጥተናል፣ ሙቀት ሰጥተናል፣ እርጥበት ሰጥተናል። but the difference is the one is found, one is kept inside the tomato juice, the other one is kept within the tomato, within the distilled water ማለት ነው። ስለዚህ እንደሱ አድርገን እንደዚህ እናስቀምጣቸዋለን ማለት ነው። አሁን control variables ምን ይሆናል ማለት ነው? control variables, the control variables, the factors that just already helps the seed to germinate. That means, you know, the worms, the air, and the, what do you call it, you know, the water. Those things are known as the control variable. Confounding variable ብለን የምንጠራው ደግሞ ነገር አለ እዚህ ጋር። confounding variables ምንድነው? ይሄ variable so ይሄ confounding variables ብለን የምንጠራው ደግሞ ከኛ ካፓሲቲ በላይ ነው። አሁን ሙቀትን መወሰን ይችላል፣ አየርም መወሰን ይችላል፣ እርጥበትን መወሰን ይችላል። እንደገና ደግሞ ቀደም ብለናል ቶማቶ ጁስ ማድረግ ይችላል፣ ዲስትሪቢዩተር ውስጥ ማድረግ ይችላል። ኢንዲፔንደንት ቫሪያብሎች ኮምፓውንዲንግ ቫሪያብልን ግን እኛ አፌክት ማድረግ አንችልም። እሺ እኛ ኮንትሮል ማድረግ የምንችለው ነገር አይደለም። ለምሳሌ አዲስ አበባ ላይ ጠዋት ላይ ቅዝቃዜ ሊፈጠር ይችላል፣ ጠዋት ላይ ሙቀት ሊኖር ይችላል። ጠዋት ያለው ሙቀትና ጠዋት ያለው ቅዝቃዜ እኛ የምንወስነው ነገር አለ። ያ ኔቸር በሚፈጥረው ነገር እሺ ያ ነገር አፌክት ያደርገዋል። ሲድ ጀርሚኔሽኑ አፌክት ያደርገውም እንደው ነው but that's beyond our capacity. እሺ እሱን ማቆም አንችልም። ሙቀቱን ማሞቃን እንዲሞቅ ማድረግ ሁሌ እንዲሞቅ ማድረግ አንችልም። ቅዝቃዜ እንዲኖር ማድረግ አንችልም፣ ወይም ቅዝቃዜ እንዳይኖር ማድረግ አንችልም። ስለዚህ እንዲህ አይነት ፋክተሮች ከኛ አቅም በላይ የሆኑ ፋክተሮች ምን ተብለው ይወሰዳሉ? በቀጥታም በተዘዋዋሪ እዚህ ዲፔንደንት ቫሪያብል ላይ ኢፌክት አላቸው። ግን ከኛ አቅም በላይ የሆኑ ነገሮችን ምን ብለን እንጠራቸዋለን? ኮምፓውንዲንግ ቫሪያብልስ ማለት ነው። በቀጥታ አፌክት የሚያደርገው ነገር ኢንዲፔንደንት ቫሪያብል፣ አፌክት የሚደረገውና ሜዠር ማድረግ የፈለግነው ነገር ዲፔንደንት ቫሪያብልስ። ኮንትሮል ደግሞ የምንላቸው እኛ ኮንትሮል አድርገን ለሲዱ ኢኳሊ አቬሌብል ያደርግ ነገር አለ። for both seeds, those which is kept inside water and those which is kept inside, you know, the tomato juice ማለት እንደዚህ በማድረግ እነዚህን አጀስት እናደርጋለን። ስለዚህ ይሄን በማድረግ እንግዲህ ይሄንን ምሳሌ በመውሰድ ኮንትሮል ቫሪያብልስ፣ you know, dependent variables, independent variables እና ኮምፓውንዲንግ ቫሪያብልስ ሌሎች ምሳሌዎች አሉ። አሁን በዚህ ላይ መጨመር ስለማልችል ክላስ ላይ በለጠ እናወራቸዋል ብዬ አስባለሁ። እንግዲህ አሁን ላይ ያለኝ ሰዓት አልቋል። ቀጣይ ፊዚክስ ስላላችሁ እዚህ ጋር ማቆም ይኖርብናል ማለት ነው። ምናልባት ጥያቄ ካለ አንብቡትና ኔክስት ታይም ክላስ ከመጀመራችሁ በፊት የተወሰነ ጥያቄና መልስ እናደርግ እንችላለን። so next time we'll talk about, you know, the biology and HIVs እነሱ ላይ የበለጠ ደግሞ ዲስከስ እናደርጋለን። ከዛ በፊት ያሁኑ ፓርት ሜቢ የተወሰነ በሪቪዥን መልክ እናያለን ማለት ነው። so thank you for today. Let's better stop here. Okay.